Good morning, all. Today we study the topic of enzyme nomenclature and enzyme inhibition. In the previous lectures, we study the factors affecting on the enzyme activity is the temperature, pH, substrate concentration, concentration of enzyme. But we only remain the enzyme inhibition. In that lecture, we cover that, and after all the we understand the classification and the nomenclature, how the names of the enzymes and the code of the enzymes uh, given by the enzyme commission to the enzymes, right? Nomenclature or the enzyme classification, the IUBMB has recommended system of the nomenclature of the enzyme, and according to them each enzyme is assigned with two names: travel name and systemic name. Systemic name is the each enzyme is characterized by the code number. Enzyme commission number, EC means enzyme commission number, and contain a 4-digit separated by dotted. The number is assigned to each enzyme representing the classes, subclasses, subclasses, and the individual enzyme in this number. First digit represents the class of the enzyme. Second digit stands for the subclass of the enzyme. Third digit stands for the subclass or subgroup of that enzyme. Fourth digit gives the serial number of the particular enzyme in the list. For example, EC2.7.1.1 for hexokinase. This shows the EC means enzyme commission number, 2.7.1.1 for the hexokinase. According to the IUPMB system of the enzyme nomenclature, enzymes are grouped into the 6 major classes. In 1964, the IUB system of the enzyme classification has been in force. Enzymes are divided into 6 major classes. Each class on its own represents the general type of reaction brought about by the enzyme of that classes. First is the oxidoreductase. Oxidoreductase is the enzyme involved in the oxidation reduction reaction. We learn in the previous lectures also. Only I remind these enzymes how the function they occur. Second is the transferase, enzyme that catalyze the transfer of the functional group. Hydrolases, enzyme that bring about the hydrolysis of the various compounds. Right. Lyases, fourth number is the lyases, enzyme specialized in the addition or removal of the water, ammonia, CO2, etc. Isomerases, enzyme involved in all the isomerization reaction. And sixth is the ligases, enzyme catalyzing the synthetic reaction where two molecules are joined together and the ATP is used. Correct. This is the classification. Factor affecting the reaction, only the inhibitor are remaining to study in the factor affecting. Enzyme inhibition. What is the enzyme inhibition? Any substances that can diminish the velocity of the enzyme catalyzed. Inhibitor of the enzyme is the important number of compounds have the ability to combine with a certain enzyme in either reversible or irreversible manner and thereby block catalysis by that enzyme. Such a compound are called as inhibitor. What is the inhibitors? Which can diminish the velocity of enzyme catalysis, right? The enzyme inhibitor decreases the activity of an enzyme without significantly disrupting their dimensional macromolecular structure. Enzyme inhibition provides a valuable information on the mechanism of the power of the enzyme catalysis, substrate specificity, nature of functional group at the active site. These includes drug, antibiotics, poisons, and the antimetabolites. Useful in understand the sequence of the enzyme catalyzed reaction, form the basis of the drug designing. The types of enzymes are the reversible inhibitors and irreversible inhibitors. These two are the inhibitors types. In that, the reversible inhibitor can be classified into another three types: competitive, non-competitive, and uncompetitive. In irreversible inhibitor forms the covalent bond with the specific function, which may in some manner be associated with the catalytic activity of the enzyme. You understand that the inhibition is incomplete at the first but continuously increase with the time because the chemical modification of an increasing the fraction of the enzyme molecule takes place. It gives important information about the identity of the catalytic functional group at the active site. There are many examples of the enzyme inhibitors which covalently not bind at the active site but physically block the active site. You understand that? The inhibitor cannot be released by the detection or dialysis or kinetically. The concentration and hence the velocity of the active enzyme is lowered in proportional to the concentration of the inhibitors, and this effect is that of the non-competitive. You see the competitive inhibition, right? Here is the figure. Substrate first substrate and enzyme complex. In first figure, green color substrate is there, and competitive inhibitor is the pink color. The enzyme and the competitive inhibitors complex is there. As the name suggested, inhibitor of this type compete with the substrate for the same active site. Competitive inhibition can be reversed by increasing the substrate concentration. Usually, the inhibitors have a molecular structure similar to those of the substrate. Both inhibitor and substrate cannot associated with the site at the same time. The inhibitor can combine with the free enzyme in such a way that competes with the normal substrate binding at the active site. Right? In competitive inhibitor reacts reversibly with enzyme to form enzyme inhibitor complex analog to ES complex. See this. The inhibitor molecule is not chemically changed by the enzyme following the Michaelis-Menten formation. For example, competitive is the inhibition of the succinate dehydrogenase. In that case, if the sufficient substrate or increase in the substrate concentration, usually inhibitor have a molecular structure similar to those the substrate. Both inhibitor and substrate cannot associate with the site of the same time. Correct. In non-competitive inhibition, Non-competitive inhibitor can combine with either the free enzyme or the ES complex, interfering with the action of both non-competitive inhibitor bind to the site on the enzyme. Other than that, the active site of often to deform the enzyme so that it does not form the enzyme substrate complex at its normal rate, and once it forms enzyme substrate complex do not decompose at the normal rate to yield the product. Is that clear? These effects are not reversed by increasing the substrate concentration. In the non-competitive inhibition, the reaction is the inhibitor yield to inactivate form of EI and ESI in enzyme inhibitor and enzyme substrate inhibitor complex. Right. Now, uncompetitive inhibition. Uncompetitive inhibition in this inhibitor do not combine with the free enzyme or affect its reaction with the normal substrate. However, it does not combine with the ES complex and to give an inactive ESI complex which cannot undergo further the reaction to yield a normal product. This relationship shows the degree of the inhibition may increases when the substrate concentration is increased. Right? Not overcome by increasing the substrate concentration. Both the Km and the Vmax are reduced. In uncompetitive inhibition binds only the enzyme substrate complex, do not have the capacity to bind to the free enzyme. You understand that? See the difference between the competitive, non-competitive and uncompetitive inhibitors plots in these graphs. Okay.